So hello students and welcome back again to yet another video tutorial. In this video tutorial we would be talking about the axis development in the case of Drosophila melanogaster. That how does the axis development in the case of Drosophila melanogaster takes place. So Drosophila melanogaster has been used as a model organism because of several key important features which are being associated with Drosophila melanogaster. These key important features include the following. First, the fruit fly are comparatively smaller in size and can grow effectively and easily in the laboratory. As these are much more easy to grow in the laboratory, so they have been extensively used. They are relatively small, effectively easy to be maintained and to be analyzed in the laboratory. It has a generation type of only 2 weeks and produces many different types of offspring. So it has a very short generation time, and then because of its short generation time, it has the ability to produce many offsprings. That is why it has been considered as one of the best model organisms. Embryo develops outside the mother body. Okay, so the embryo is always going to develop outside the mother body, so that's an important thing. And in addition, there are vast amount of information on its genes and other aspects of its biology. See, it was Drosophila melanogaster which was almost the first organism to be entirely sequenced. ड्रोसोफिला मेलानोगैस्टर ही वो सबसे पहली ऑर्गेनिज्म थी जिसकी पूरी सीक्वेंसिंग की गई, जिसकी वजह से आज हमें सारी इन्फॉर्मेशन है ड्रोसोफिला मेलानोगैस्टर के बारे में। क्योंकि हमारे पास में सारा इन्फॉर्मेशन है ड्रोसोफिला मेलानोगैस्टर के बारे में, दैट इज वाई वी आर गोइंग टू रिलेट ईच जीन एंड इट्स फंक्शन along with the functionality of each genes in the case of humans. Drosophila melanogaster was the first organism through which we were able to understand the embryonic development, how it takes place in the case of humans. एम्ब्रायनिक डेवलपमेंट क्या होता है, किस प्रकार से होता है, किस तरीके से होता है, इस बात को पता लगाने के लिए भी जो सबसे पहला ऑर्गेनिज्म था ना, वो कौन था? दैट वन वाज ड्रोसोफिला मेलानोगास्टर. सो लेट अस लुक एट ड्रोसोफिला मेलानोगास्टर एज अ मॉडल ऑर्गेनिज्म. What have been its contribution, why it is being used. Drosophila ovariole oogonium divides into 16 cells. The one is referred to as an oocyte, and there are 15 nerve cells, and all these cells are interconnected with regard to each other. So what you are going to find here is that you are going to find there is an apex, there is the cytoplast, a layer of cells is present, clones of cystocytes. Cystocytes are the cells which will further divide and will lead to the formation of nerve cells. ये वो सिस्टोसाइट्स क्या होती हैं? प्राइमरी सेल्स होते हैं जिनके अंदर लगातार डिवीजन होता है, एंड एज दीज सेल्स undergo spontaneous division within themselves, so it is these cells that will give rise to the nerve cells. These are the cells that will produce the nerve cells, so they are the one of the most important cells that are present, and they are called as cystocytes. Okay, so these cystocytes divide and they will lead to the formation of follicle cells. Once they form the follicle cells, then follicle cells, see this, these are the follicle cells, and inner side the cystocytes are going to divide. As the cystocytes divide, they will be forming the nerve cells. cystocytes divide करते चले जाते हैं और nerve cells को बना देते हैं. अब बेटा nerve cell का काम क्या होता है? जैसे देखो nerve cell, आप इसे ऐसे समझिए, ये एक ऐसे cells हैं जो mRNA को बनाते हैं, जो प्रोटीन को बनाते हैं, और प्रोटीन को बनाने के बाद में mRNA को बनाने के बाद में साइटोसोल ऑफ द एग में एक्यूमुलेट करा देते हैं. Nerve cells are the one which are going to accumulate the proteins and mRNA into the cytosol or the cytoplasm of the egg cell. Okay, once it would be accumulated in the cytosol, once it would be accumulated in the cytoplasm, then what is going to happen? Then from the cytoplasm it can be effectively used. इसके बाद में cytoplasm के अंदर ये mRNA को accumulate करा देंगे जो anterior posterior dorsal और ventral polarity को determine करेगा. It would be the one that would be able to determine the anterior posterior dorsal and ventral polarity of the Drosophila melanogaster. Drosophila melanogaster की सारी polarities का जो division है वो यहीं के थ्रू होता है, इन mRNAs के थ्रू ही होता है जो nerve cells produce करती हैं. Now we must understand that how does the axis development. Axis development means that in an organism there are 4 different features. One is referred to as the frontal part, is referred to as the anterior part. So for this model of dog, this is the anterior part, this is the posterior, this is the dorsal, and this is the ventral part. So in this lecture we have to understand that what will happen in the case of a fruit fly, and how does the anterior, posterior, dorsal and ventral part of the fruit fly are being produced, that in what manner all these different components of the fruit fly are being produced. You can look here, both of these axes, whether it is the anterior or the posterior, are established very early in development, and in some species even before fertilization. The most important of the point is this, that the axis development in the case of Drosophila melanogaster is in such a manner that it is being determined even before the act of fertilization, that the axis development is going to take place and it is going to be determined. Now there is the axis that is present, one is referred to as the anterior axis and the posterior polarity. The anterior and the posterior polarity of the Drosophila is being determined even before fertilization with specific type of genes which are referred to as maternal effect genes. जो genes इनको बारे में हमें बताते हैं, those genes are referred to as maternal effect genes. What are those genes? Those genes are called as maternal effect genes. Now how does these maternal effect genes influences the development of different poles in the case of Drosophila melanogaster? You can look here, anterior and posterior polarity of the embryo, the larva, and adult has its origin in the anterior and posterior polarity of the egg. Now, what do you mean by maternal effect genes, and how does these maternal effect genes are going to be influenced? The maternal effect genes expressed in the mother ovaries produces mRNA that are placed in different regions of the egg. Okay, so maternal effect genes are expressed in the mother ovaries, produces different mRNA that are placed in the different regions of the egg. So while the embryonic development is going to take place, please look at the diagram shown to you that I am making. During the course of embryonic development, this is the egg that is going to be produced. Now, before the process of fertilization has taken place, there is an accumulation of an mRNA that is going to take place in this egg. And this mRNA is going to be accumulated at different concentration within the egg cytosol. mRNA का अलग अलग concentration हो सकता है egg के cytosol में. This is the cytosol of the egg. Okay, cytosol of the egg is present. These mRNA encodes transcription and translation regulatory proteins that can activate or repress the expression of certain zygotic genes. So when the sperm is going to arrive here, okay, this is not a meteor, this is a sperm, okay, moving towards the egg. When the sperm collides with the egg and fuses with the egg, then it will lead to the formation of a zygote. इसके बाद में ये zygote बना देगा. Then it will lead to the formation of zygote. Okay, once a zygote is going to be produced, then the zygotic genes are going to be expressed, and the expression of these zygotic genes will be dependent upon zygotic genes, and their expression will depends upon certain genes. Which are those genes that will be expressing these zygotic, regulating these zygotic genes? Those genes are maternal effect genes. Maternal effect genes ही इन zygotic genes को influence करेंगे, इनको regulate कराएंगे. Okay, these mRNA encodes transcription and translation regulatory proteins. Okay, so just try to understand this particular point. These mRNA encodes transcriptional and translational regulating proteins that can activate or repress the expression of certain genes. So certain genes can be activated, some of the genes, other genes can be effectively repressed. कुछ जीन्स का एक्टिवेशन हो सकता है, कुछ जीन्स का रिप्रेशन हो सकता है, और ये जीन्स अलग अलग तरीके से वर्क कर सकते हैं. सो ऑल दीज जीन्स आर गोइंग टू वर्क इन डिफरेंट मैनर. Mutations of these genes produces no effect on the mother. Instead, its effect has been seen in the next generation. That means the mutant phenotype. So mutations have been reported to be present in the case of mutant phenotypes. These genes are called as mutant. These genes are the maternal genes, and corresponding genes are called as maternal effect genes. So these genes are going to influence the type of offspring that is going to be produced, and these genes are commonly referred to as the maternal effect genes. Okay, Deleting annotation and moving to the next slide. What we found on the next slide is that we find here that four maternal effect genes are very important. Which are the four maternal effect genes? Bicoid, hunchback, nanos, and caudal. And how the expression of these genes is going to take place, that we have to understand. Are critically important for the formation of anterior posterior axis. Among these, bicoid and hunchback regulate the production of anterior structure. Nanos and caudal regulate the formation of posterior structure of embryo. These four genes are transcribed in ovarian nurse cells, cells that support growth and development of the oocyte. The hunchback and caudal mRNA transported to oocyte where they uniformly distribute in the cytoplasm. देखिए सबसे पहले oocyte का जब फॉर्मेशन होता है उस समय यहां पर चार तरीके के जीन्स, फोर जीन्स आर बीइंग ट्रांसक्राइब्ड बाय द oocyte and their mRNA is accumulated in the oocyte cytosol. Which are these four different types of genes? These four different types of genes are the bicoid, hunchback, nanos, and caudal. And once they are being expressed, then they will be used in order to form the anterior and posterior polarity of the Drosophila melanogaster. Bicoid mRNA are located in the anterior part of the oocyte and bound to the anterior microtubules through their 5' untranslated region. You know that in an mRNA, supposedly this is an mRNA. In an mRNA, there is a 5' cap. There is a cap-like structure that would be present called as 5' capping. There is a poly tail which is present, and then there are certain regions which are present. For example, 5' UTR, 3' UTR. Then it is the binding of the bicoid mRNA is going to take place through the 3' UTR. Nanos mRNA are located in the posterior and are bound to the posterior microtubules. Nanos mRNA posterior side में होते हैं and they are bound to the posterior microtubules. How? That we have to look into the following diagram. Now see, dear students, now what we find here is that how the expression of the hunchback and caudal proteins as well as the bicoid and the nanos proteins are going to be varied. Now firstly, we must talk about the mRNA molecules. You can look here, the hunchback and caudal mRNA are being expressed as such throughout the egg. Throughout the egg cytoplasm, the caudal and hunchback mRNA are expressed at a particular level. But what you found in the case of bicoid and nanos mRNA expression, bicoid mRNA is always being expressed in an area which is referred to as the anterior pole. Anterior pole, the mRNA which is going to be expressed is referred to as bicoid mRNA. This is the bicoid mRNA. On the case of posterior part, the posterior part, the mRNA which is going to be expressed is referred to as the nanos mRNA. And both of these nanos and bicoid mRNA are being accumulated in the cell, or all of these four mRNA are being accumulated in the cell, in the egg, even before the process of fertilization. ये चारों mRNA egg के अंदर fertilization से पहले ही रख दिए जाते हैं, accumulate करा दिए जाते हैं अलग अलग साइट पे. Once the bicoid mRNA is going to be present, then how the bicoid protein is going to be formed, that we have to look. So you can look here, this is the bicoid mRNA and this would be the level of the protein that would be produced. This is how bicoid protein will be produced from anterior towards posterior in a decreasing manner. Similarly, you can find the nanos mRNA is here. So nanos protein will be produced in this particular orientation. Okay. Then you can find here is that nanos support another type of protein that is referred to as caudal. Once the nanos protein is going to be produced, it is going to support the caudal. It acts as a positive modulator for caudal. Which protein acts as a positive modulator? Nanos protein. Nanos protein. Nanos protein caudal का positive modulator है. So once it is going to activate caudal, then how the caudal expression is going to be altered? Caudal is going to be expressed in the presence of the nanos protein to its highest level. Okay. And bicoid protein is the negative modulator of caudal. Bicoid is the negative modulator of caudal. But bicoid is the positive modulator of a protein called as hunchback. It is a positive modulator of hunchback, and nanos is a negative modulator of hunchback. What is nanos? Nanos is a negative modulator of hunchback. That means where this hunchback is going to be expressed, hunchback is going to be expressed towards bicoid. So this is the ratio of expression of the different genes, and because of the expression of these genes, the Drosophila embryo gets differentiated into head, thorax and abdomen. इन्हीं जीन्स के प्रोडक्ट्स के एक्सप्रेशन की वजह से ड्रोसोफिला का एम्ब्रायो हेड, थोरैक्स और एब्डोमेन में डिवाइड हो जाता है. नेक्स्ट पॉइंट.
Today we will learn about a basic topic in developmental biology, the anterior-posterior axis formation in Drosophila. This is an adult. Today we will learn about a basic topic in developmental biology, the anterior-posterior axis formation in Drosophila. This is an adult fruit fly that is Drosophila melanogaster, in which the anterior to the posterior axis is shown. The anterior-posterior axis is specified during oogenesis. The oocyte moves into the posterior region of the egg chamber, while nerve cells fill the anterior portion. Now, coming to the oocyte region, let's zooming into the posterior part. The oocyte nucleus moves toward the terminal follicle cells and synthesizes gherkin proteins. The terminal follicle cells express torpedo, the receptor for gherkin. When gherkin binds to torpedo, the terminal follicle cells differentiate into posterior follicle cells and synthesize a molecule that activates protein kinase I in the egg. Protein kinase orients the microtubules such that the growing plus ends are at the posterior. Bicoid mRNA binds to dynein, a minus-directed motor protein associated with the non-growing end of microtubules. Dynein moves the bicoid mRNA to the anterior end of the egg.
Now, what we have to study here, please look here. See, you know that there is the anterior part of the Drosophila, there is the posterior part of the Drosophila. The formation of the anterior part and posterior part typically depends upon the maternal mRNA which is going to be present. So the maternal mRNA which is going to be present is going to differentiate the anterior part and the posterior part from each other. Okay, so what we have to study here is that here what you have to find is that this is the anterior part, okay, of the Drosophila melanogaster, and this is the posterior part of the Drosophila melanogaster. Then how these proteins have been produced. Now see, this is the embryonic development of the Drosophila melanogaster. This is the cellular blastoderm. The blastocele cavity has been completely filled with the division of cells, and now this is a cellular blastoderm. Blastocele cavity जो खाली cavity थी वो cellular blastocele में convert हो चुकी है. अब ये जो सेलुलर ब्लास्टोडर्म कैविटी यहाँ पर बन चुकी है, इसके अंदर अलग अलग सेल्स डिफरेंशिएट करेंगे. यहाँ पर आपको एक स्पेशलाइज्ड सेल्स मिलेंगे. दीज सेल्स आर रेफर टू एज द नर्व सेल्स. देन आफ्टर द डिफरेंशिएशन ऑफ द नर्व सेल्स, लेट अस लुक हियर. These are the nerve cells that are forming the maternal mRNA. There are uncommitted follicular cells. Uncommitted follicular cells are the cells that are not fully differentiated. The differentiation of any cell depends upon a particular type of protein which is present, okay, and that type of a protein which is present is commonly referred to as the gherkin mRNA. Gherkin mRNA, which is also referred to as gherkin protein. I've already told you, you just look here. These are the terminal follicular cells which are present. There is a nucleus of the oocyte which is present. Now the nucleus began to synthesize an mRNA, and that mRNA is referred to as gherkin mRNA. न्यूक्लियस क्या बनाता है बेटा? एक mRNA बनाता है जिसका नाम है gherkin mRNA. ये gherkin mRNA एक प्रोटीन के स्ट्रक्चर के ऊपर बाइंड करेगा, and it is the structure or the receptor for the gherkin protein that is referred to as the torpedo. Torpedo is referred to as the receptor of the gherkin protein. It is this torpedo where the gherkin receptors are present. Once the gherkin binds to the torpedo, it will cause the posterior follicular cells to differentiate. यानी कि किसी भी सेल का डिफरेंशिएशन बेटा कब होता है? जब बाहर से उसके ऊपर सिग्नलिंग मॉलिक्यूल आएगा. बाहर से सिग्नलिंग मॉलिक्यूल आएगा, सेल को डिफरेंशिएट करा जाएगा. And which is the signaling molecule that is present? The signaling molecule is referred to as the gherkin protein, and the receptor is referred to as the torpedo receptor. Gherkin protein binds to torpedo receptor, causes the differentiation of the cells to take place. Okay, once the differentiation of the cells is going to take place, then the posterior cells have been fully differentiated. Okay, so these are the cells that have become fully differentiated. So then we have seen that the gherkin protein was binding to the torpedo receptors and causing the differentiation of the cells. Once the differentiation of the cells takes place, then you find that this is how the cell appears. The developing egg appears. And in this case, you find here that bicoid mRNA will be transferred from the central part and accumulated towards the posterior end. How?
Protein kinase orients the microtubules such that the growing plus ends are at the posterior.
Okay, so there is the plus end which are present towards the posterior part. There are negative ends which are present towards the anterior part. Then there are specific proteins which are going to be expressed called as kinesin and dynein protein.
Bicoid mRNA binds to dynein, a minus-directed motor protein associated with the non-growing end of microtubules. Dynein moves the bicoid mRNA to the anterior end of the egg.
अब देखिए बाइकोइड एमआरएनए को एंटीरियर एंड पे ले जाने वाली जो प्रोटीन है उसका नाम क्या है? डायनिन. डायनिन प्रोटीन इज अ माइनस एंड डायरेक्टिंग मोटर प्रोटीन. प्लीज मूव टू सेल बायोलॉजी इन योर बुक. Okay, and there you will find that dynein protein is a minus and directing protein, minus and directing motor protein. It will move over the microtubule, will cause the bicoid mRNA to be transferred towards the anterior side. Similarly, Oscar mRNA, you remember the Oscar mRNA? It is going to be transferred towards the posterior side on the basis of another type of protein called as kinesin. Kinesin will take this Oscar mRNA, this is the kinesin, and will transfer it into the posterior side.
The Oscar mRNA becomes complex to kinsin 1, a plus-directed motor protein that moves it toward the growing end of the microtubules at the posterior region, where Oscar protein can bind nanos mRNA.
Okay, so Oscar protein can bind with nanos and mRNA. And here there is the bicoid protein which has been produced. So this is the bicoid mRNA that is going to be transferred towards the anterior spine.
The nucleus, with its associated gherkin proteins, migrates along the microtubules to the dorsal anterior region of the oocyte,
Okay.
and induces
Now-
the adjacent follicle cells to become the dorsal follicle cells.
Now it is the movement of the nucleus that is going to take place, and it will cause the adjacent follicular cells to get converted into dorsal, whereas the other will get converted into ventral.
And thereby finally the axis formation is completed.
This is how axis formation is going to take place in the case of Drosophila melanogaster. Just a quick revision that during the embryonic developmental stages there are nurse cells, there is the oocyte, then in the oocyte there is the prominent nucleus which is present, which forms an mRNA of the gherkin mRNA. Gherkin mRNA gets converted into gherkin protein. Gherkin protein binds to a specific receptors called as torpedo. Torpedo receptors are the one which causes the terminal follicular cells to differentiate. तो न्यूक्लियस से निकलती है बेटा गुरकिन प्रोटीन बाइंड करती है टॉर्पीडो से जब बाइंड करती हैं तो न्यूक्लियस के साथ में सेल्स डिफरेंशिएट हो जाते हैं. यहाँ से आपको पता होगा पोस्टीरियर फॉलिकुलर सेल्स कुछ प्रोटीन्स को सीक्रेट कराते हैं. It is these proteins which are going to be secreted which will lead to the formation of microtubules. So kinesin and dynein are going to work in antagonistic manner. Kinesin is a plus end directed motor protein. Dynein is a minus end directed motor protein. So kinesin is going to take away the Oscar towards the posterior side, causing the formation of the germ cells towards posterior side, whereas dynein will cause the accumulation of bicoid towards the anterior side. So what we have is that we have bicoid mRNA in association with exemperentyl and dynein proteins accumulate towards the anterior side. Oscar mRNA has been accumulated towards the posterior side. So bicoid binds to dynein, moves on a non-growing minus end of microtubule, and Oscar is going to bind with kinesin. Oscar binds with nanos mRNA, move towards growing plus end of the microtubule. Okay, this is the concentration gradient that is going to be produced. Okay, so this is what we have seen. That now what we have to study is about other different types of protein, that is referred to as smung proteins and nanos and bicoid proteins, how they are going to be produced. So this is what we would be studying in our next lecture. Hope you have enjoyed the lecture. That's all for today. Meet you tomorrow at same timing. Thank you. Good night. Bye bye.